Hello, everybody, and welcome to today's webinar. My name is Olga Bodie, and I'm a SYNGAP parent and volunteer with SYNGAP Research Fund. Our talk today discusses the use of milk exomes to increase the expression of SYNGAP1 in SYNGAP1 mice. I have the pleasure to introduce today's speaker, Dr. Janusz Zempliny. Dr. Janusz Zempliny earned a PhD in nutrition sciences from the Justus Liebig University in Germany and received postdoctoral training in pediatric nutrition, nutritional biochemistry, and molecular and cell biology in different institutions. Dr. Zempliny joined the University of Nebraska-Lincoln in 2001, where he rose through the ranks and now is a Willa Cather Professor of Molecular Nutrition and founding director of the NIH-funded Nebraska Center for the Prevention of Obesity Diseases through Dietary Molecules. The Zempliny Lab has a long track record of research in nutrition and epigenetics. His laboratory is interested in natural nanoparticles that play an important role in cell-to-cell communication and the regulation of metabolism. Currently, the Zempliny Lab is focusing on the role of exomes in human milk in the neurological development in infants, interactions between milk exomes and the gut microbiome, and the use of milk exomes in the delivery of drugs. A recorded version of this webinar will be available on the SRF website under webinars under the family menu. By the end of the presentation, you will have the opportunity to get the answer to your questions. We'd love to hear from you. Please write your question in the Q&A below. For those of you just joining us, welcome. And again, our speaker is Dr. Janusz Zempliny, and his talk is investigating the functional single-cell biology of SYNGAP1 pathways. It's now my pleasure to turn things over to Dr. Zempliny.
Thank you, Olga. That was a very nice and kind introduction. Before I start my presentation, I would like to make your group a compliment. I've interacted with many funding agencies throughout my career, but your group has been the most pleasant to interact with by far. What comes to mind when I think of your team is you have a very important objective, you have a knowledgeable and invested leadership, you're very unbureaucratic, which I really like, and you are just pleasant people to interact with. Then I also have one housekeeping item. Our university has installed motion sensors in our office, so if I, in my presentation, jump up and wave my arms, this is not because I'm going crazy, it's just to get the lights back on. This may or may not happen to my presentation. So let's get started. My university requires me to disclose any potential conflicts of interest. There really is not, turn on my laser pointer here. There really is not a whole lot to show for. It's just my funding agencies. And I do serve as an advisor to a Boston-based company named PureTech Health. They are also in milk exosomes and drug delivery, with the focus on brain cancer. So it's really not relevant, not directly relevant to what you and I are working on. When I was invited to give this presentation, SYNGAP parents and leadership informed me that the audience will be very diverse. So some of you are scientists, some of you are parents, some of you are donors, and then maybe just lay public who wants to chime in and then tune in and then listen to this presentation. So I'm trying to, or was trying to set the tone in my presentation for not losing anyone on this talk. So basically catering to the educated lay public, I think. So let me start off by telling you a little bit about extracellular vesicles. That's a group of compounds that encompasses a variety of components. The ones that we are interested in are exosomes, and they're spherical particles with the size of a virus, so about 100 nanometers, so really tiny particles. Then there's another class called microvesicles. They're a little larger, up to 1 micrometer, which is the equivalent of a bacterium, and these are secreted by healthy cells. There's also apoptotic bodies. They're much larger, 5 micrometers, and they are shed by cells when they die, so they're really not relevant in healthy biology. And for reference, the cell, the mammalian cell, typically is about 10 micrometers large. So exosomes, well, again, there was two major classes that of biological relevance. Microvesicles, which we are not going to focus on today because they just flap off the plasma membrane. It's tough to control their cargo, and their role in cell-to-cell communication is not so well understood. We are going to focusing on exosomes. They are produced in cells, in the cell interior, in exosome secreting or donor cells, released into the extracellular space. So this is basically your bloodstream. And then they travel to adjacent cells, neighboring cells, short distances, or can travel very long distances, for example, from the liver to the brain. In our case, we're talking about even longer distances because we are talking about exosomes in milk being absorbed and traveling to tissues. Once these exosomes reach the recipient cells, they either bind to the cell surface and trigger all kinds of cellular responses, or they internalize by fusing with the cell membrane and release their cargoes into the cytoplasm of the cell, or they're internalized and then later release their cargoes. When I say cargoes, you can see an exosome right here. It's crazy what they hold. There's lots of RNA, there's DNA, there's proteins, there's lipids. It's really amazing for these tiny particles what all they can hold and what they deliver to these recipient cells to change the metabolism in recipient cells. So cartoons are nice. This is an image of a real cow's milk exosomes from an electron microscope. You can see they're about, this is 200 nanometers, so they're about 100 nanometers in size. And this is how they look like in real life. So research in my lab, Olga alluded at this a little bit. I think of having two streams of research, both related to milk exosomes. One of them is the delivery of drugs and therapeutics. This is what your group is interested in. And the other stream of research is really milk exosomes, human milk exosomes in infant nutrition, and cow's milk exosomes in adult nutrition. Both streams confluent, merge basically at the interest or the shared interest of promoting human health. So the project aim, when I submitted my proposal, was basically stated as assess whether milk exosomes deliver SYNGAP, mRNA, and blah, 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 blah. And I promised at the beginning of my presentation, I will keep this very comprehensible and simple. So this big aim can be condensed to just saying, well, we want to increase SYNGAP in the brain. And this aim of the proposal really addresses a problem that the SYNGAP patients have. So there's 2 copies of the SYNGAP gene in a brain. And in SYNGAP patients, you have 1 functional copy, the solid line here, half circle, and then you have 1 copy that does not function. This is the white part of the circle. In normal individuals or healthy individuals, you have 2 functional copies of the SYNGAP gene. And what we want to achieve in this project is deliver DNA that codes for SYNGAP through milk exosomes to the brain and basically restore the activity, the missing activity from this copy to just achieve a normal level of SYNGAP in a brain and thereby make brains healthy. So now you wonder, why should this work? Isn't this a crazy idea? Milk exosomes, these nanoparticles in milk for treatment of SYNGAP. Some of you may remember high school biology or college or graduate school biology, and you may ask, well, these exosomes or anything that enters the gut, the gastrointestinal lumen is degraded very rapidly, either by gut microbiota or by enzymes that are secreted in the gut. And what you can see here, it's just a reference to 3 papers that showed exosomes are really, really stable. They are not degraded in the gut lumen. And that is really an important premise for what we are doing here. started with a group in Japan in 2012. That idea was copied by a group in Toronto, Canada, and then again copied, and I say copied, not stolen, but copied by a group in California. They all found the same thing. These exosomes and their cargos are very, very stable in the GI tract. And then we added a piece of information by showing that even when you process milk and milk exosomes at an industrial level, for example, homogenizing milk, heat sterilizing milk, adjusting the fat content of milk, and so on, these exosomes and their cargos are very, very stable. So we really don't lose exosomes in the GI tract and when processing exosomes at an industrial scale. So first data piece that I'm going to show you now. In our lab, we created what we call an exosome and cargo tracking mouse. It's an extremely complicated story, so I'm going to spare you all the details. But one thing I have to tell you, so if this is an exosome produced by this mouse, it is endogenously labeled with fluorescent proteins. So in other words, these exosomes by this exosome cargo tracking mouse, they emit light, very strong light, which allows us to track these exosomes in tissues. In this particular experiment, what we did in order to demonstrate absorption of milk exosomes was to foster normal pups, I mean wild-type pups that do not express these exosomes, to a dam mom that expresses these fluorescent exosomes. And then in a controlled experiment, we fostered these normal pups to a normal dam that does not express these labeled exosomes, where these mice nurse for 17 days. So normally mice are weaned at about 21 days. So we wanted to achieve a fairly long exposure to these milk exosomes, but stop the study before these little pups transition slowly to solid foods. So 17 days is right at the end where they exclusively rely on milk. At that point, we harvested the tissues from these pups and the control pups and looked at the light in these tissues. And now it's important to remember these normal pups, again, they cannot make fluorescent exosomes or any fluorescent protein. So any fluorescence light that you will see in these tissues would have to come from mom's milk, because this is the only thing that these pups had received. And then again, we dissected the tissues, and what you can see here, really a major signal in the brain, and then also quite some accumulation in the liver, a little bit in the kidney, and considerable accumulation in the intestinal mucosa, so the gut wall. In controls, you see no signal, which is what we expected. In all these tissues, just autofluorescence, I mean background noise in the gut intestinal mucosa. So this is very conclusive evidence that milk exosomes transfer from mom to pups, or that milk exosomes in general are absorbed and accumulate in tissues, including, and that's relevant for your group, including in the brain. When I think met the second time with folks from the Syngap Research Fund, one of your panelists asked me about, so mouse to mouse, that is nice, but what about milk exosomes across species boundaries? Because we are dealing with cow's milk exosomes delivering cargo, Syngap, to humans. So in other words, does this also relate to human or other species? And the answer is yes. So what we did in this experiment here, we used cow's milk and isolated exosomes from this cow's milk, loaded these exosomes with synthetic compounds, something that we chemically synthesized. In this case, it was an RNA with the name MIR375. Don't have to remember that one. Basically, just the nucleic acid with a fluorescent dye, put these labeled compounds in our milk exosomes, and gave these cow's milk exosomes to mice by oral gavage. It was an oral administration of these exosomes, and then did exactly what we had done with these other pups in the previous slide, collected tissues and measured fluorescence in these tissues. And again, for this particular compound, you see strong accumulation in liver, brain, kidney, and also the GI mucosa. This is a control. Mice that actually were given exosomes and this compound, but not the compound being included or incorporated in exosomes. You can see it's really just background noise. This one is a control experiment that I don't want to elaborate on. It basically just showed us that this compound here was stable when we measured the tissue signal, and indicating that we did not just track the label that was detached from the RNA. So basically this confirms our studies from the previous mouse experiment, saying, yeah, these exosomes and their cargos accumulate in tissues. I do understand that we, in your case, are most interested in working with downstream, at least, with human subjects, with patients, SYNGAP patients. And so we had a previous publication in which we showed or demonstrated the availability of milk exosomes in humans. So this was a milk feeding study, basically, with 5 normal adults come to the lab, had them drink various amounts of milk. It's a dose response study ranging between 0.25 liters all the way up to 1 liter. And I don't know if you're used to the metric system. So half liter is a large glass of milk. 1 liter of milk per day is pushing it. I love milk, so I can easily drink 1 liter of milk per day. Many of you may not, but I do. But this is fairly physiological doses, so we're not trying to do something that is really pushing or venturing into the gallons of milk exposure to see a tiny effect. And when you consume, or when our adults consumed these amounts of these volumes of milk, you can really see the dose response in plasma. So these, and that's the cargo that we're measuring, another microRNA. You can see the more milk they drink, the more exosome cargo appears in these subjects' plasma and returns to baseline levels after, I think, 24 hours. What you see down here, MIR1 is our negative control. This is a microRNA that is not present in milk. And since it's not present in milk, you would expect there is no increase in plasma microRNA1 level after milk consumption. So milk consumption at time 0. And obviously you don't see any change, which was reassuring to see, because then you could say, okay, this is really a specific effect to milk consumption. Okay, next question that is really relevant for this project is a challenge posed by a barrier that surrounds the brain. So the blood-brain barrier that I'm sure all of you have heard about. It really protects the brain from pathogens or any microorganism entering the brain, and also excludes certain metabolites from entering the brain. So the question for us now for the milk exosomes would be, and it goes back to the slide that I showed you early on. The question now would be, what we see here in the brain, is this inside the brain? This is where we want the exosomes to go in order to deliver SYNGAP, or is this just exosomes being in the blood vessels surrounding the brain, and both would produce the same signal in theory? And what we did in order to answer this question, in the brain versus outside the brain, was to partner with the investigator at UT Southwestern in Dallas, Dr. Denise Ramirez. She has this really fancy technology. Let me go back to one. Has this fancy technology in which she can really create 3D images of brains, and these 3D images allow her and us to exactly to conclude whether something is inside the brain or just outside the brain. So what I'm going to show you now is images from 2 brains. The one that had the milk exosomes from mom delivered to the pups, and then a control brain, which is not, of course, fluorescent. And that's what Denise did. So this is the parts of the brain from these fluorescent exosomes. You can see in various regions of the brain, there's this green fluorescence, this green light, suggesting these exosomes get into the brain tissue, so they cross the blood-brain barrier, and controls I think all of you can see this. There's really no green light, meaning the negative controls were negative. So in other words, this signal is real, and these exosomes actually make it into the brain. So now, many of you may wonder, but you showed us in these slides and this slide that these exosomes do not go exclusively to the brain. They accumulate also in liver, and maybe kidney, and so on. So isn't that a problem? Doesn't this distract from the delivery of exosomes to the brain? So what is desired is actually really exclusive localization to the brain. Not so great if exosomes go to other tissues, like the liver in this case, because of two reasons. First of all, accumulation in other tissues other than the brain, just take away from the drug that you want to get into the brain. And also in some cases, depending on what you put into these exosomes, delivery to the wrong tissue could cause adverse side effects. So you really want to get be as specific as possible and not deliver these cargoes to non-target tissues. Okay, so the solution that we have to offer to this problem is the development of smart exosomes. So each tissue has a unique, most tissues have a unique zip code. So you can deliver, for example, the zip code 0001, which is the heart, or 0005, which is the brain. I'm making up these numbers, and they're not scientific numbers. So, but these tissues have unique zip codes. And what we will do, as I will show you in a minute, we will actually slap a label, a shipping label, onto our exosome. So this is our MAC exosome with the SYNGAP DNA inside. And this shipping label or address label will deliver these exosomes to the tissue that we want it to go. And this is not just a wild idea, crazy idea. There's some publications out there that talk about all these what's called homing peptides, these shipping labels to multiple tissues. I know you will not be able to read this. This is just part of a table from this paper, and there's hundreds of zip codes or shipping labels for various tissues. So muscle, pancreas, brain, kidney, and so on. So you can just pick and choose basically from these lists which zip code or which shipping label works best for you, and then of course demonstrate that it works. So in our case, what we want to do, or what we want to use as a shipping label to get our exosomes to the right zip code, being the brain, is we will put a protein called RVG. It's rabies virus glycoprotein, but just call it RVG, on the milk exosome. And we picked RVG because of a previous publication that showed RVG increased accumulation of exosomes in the brain. So this is not milk exosomes, this is mouse exosomes or mouse cell cultures in which these exosomes were changed to have this shipping label, RVG. And here you can see the data actually. So this is a brain. These are 3 brains actually. One mouse, or multiple mice, were given exosomes with the RVG, this shipping label. You can see massive accumulation of the light in the brain. These are exosomes without the label. You can see that, sure, they do go to the brain, but not nearly as much as the exosomes carrying the RVG. And this is just a brain that was not treated, from a mouse that was not treated with any exosomes. So bottom line, massive enhancement of exosome accumulation in the brain if exosomes were tagged with this RVG shipping label. Okay, so go back to your high school and college and graduate school training in biology. You now may ask me, well, we all know, I mean, these immune cells in the body, they don't like foreign particles, especially macrophages. They eat up these particles and any other compound that they think should not be here. And of course, us delivering cow's milk nanoparticles to mice or to humans, they will be recognized as foreign particles and will be eaten up by macrophages. Last summer, we published a paper in which we identified the transporter responsible for this uptake. And we will leverage our knowledge and expertise in the field to offer a solution to fix this uptake or elimination issue that these macrophages create. So the plan for that part is to put a "don't eat me" signal on our milk exosomes. So these are peptides or proteins that signal or tell a macrophage, you must not eat me. And one such modification is a protein called CD47. So we put that on the exosomes, and this is an experiment that we have already done in cell cultures. We use our milk exosomes with and without CD47 and measured the uptake of these exosomes by macrophages. And what you can see here, these are exosomes, 100% is to control exosomes that did not have the CD47. And if we had exosomes with the CD47 on the surface, elimination or uptake by macrophages decreased by nearly 60%. So this is a massive improvement as far as avoiding macrophage uptake is concerned. So this is again a quote from this proposal that I sent to the Syngap Research Fund. I don't need to read this out in detail, but basically the plan is to engineer milk exosomes that express both RVG. Remember, this is the brain homing, so that will take the exosomes to the brain, and CD47, which is a signal that tells the macrophages, stay away, I need to go to the brain. Okay, so now the logical question coming from the audience would be, how do you tell a cow to make RVG CD47 positive exosomes? Because cows don't care. Obviously, they make milk exosomes optimized for nutrition of their calves. They're not worried about drug delivery. So the solution that we have to offer in this context is to use cells from the cow's udder, cells that actually produce the milk exosomes, and they're called mammary alveolar cells, MacT cells. So we actually generated these cells, and this will allow us to actually engineer milk exosomes that express RVG and CD47. So one step that we had to take was to make really sure that cow's milk exosomes and exosomes produced by MacT cells look alike. So they're very similar, and there's not much of a difference between these two. So we published this paper also last summer showing exactly that. So looking at the size, cow's milk versus MacT exosomes, they're just underneath 100 nanometers, so same size basically. The contours, shape, they're the same. So this is a cow's milk exosome, this is a MacT cell exosome, really look very much the same. We also looked at cargos, what do they contain? And we looked at which tissues they accumulate in, and both were very, very similar. I'm not going to show you this here. Okay, now, sorry for putting questions in your mouth, but the logical question would be, well, are these MacT cells actually amenable to genetic engineering? Can you actually change the makeup of exosomes in these MacT cells? And that's really also what we have done in this paper that I just mentioned. This is actually how the MacT cells look under a microscope. And so the question being, can we engineer these MacT cells to produce exosomes with the desired modifications? I'm going to show you a light bulb here, because that's going to be important in the next slide. So we wanted a test run to establish proof of concept, engineer these exosomes to secrete, engineer the cells to secrete exosomes that emit light. And that's what we have accomplished, as you will see. So now I have to walk you through this, these images. So what you see in the first panel, A, is normal MacT cells in a dish compared to MacT cells that we engineered to produce these red exosomes. And I think the difference is obvious and striking. So the normal MacT cells, no red production, and these transformed, engineered MacT cells, bright red. So they do express the light bulb, basically, that we want to be in there. In order to keep cells happy, now moving to panel B, in order to keep these cells happy, you have to culture them and soak them in nutrition fluids, basically. And so the next thing that we looked at was the supernatant of these cells, the media. Again, the normal MacT cell media, there was no fluorescent in the supernatant, whereas these engineered MacT cell, they secreted something in the media that produced a red light, probably our light bulb modified exosomes. Those exosomes are secreted in the extracellular space, as we said, and that's what we tested in panel C. We spun this supernatant. Now we're focused on this one here. We spun it at increasing G forces, and initial spin is 16,000 G, and the red light stayed in the supernatant, which is expected for exosomes, as you will see in a second. Then we spun even harder, 83,000 G. And again, I mean, most of the supernatant, most of the red color stayed in the supernatant, which is what we wanted. So because exosomes are tiny and they're extremely tough to really precipitate by spinning them in a centrifuge. So we increased the force, the G force, to 130,000 G. Looked at the medium, so the supernatant, and now you can see all the color is gone. We collected a pellet, which is not shown here, dissolved the pellet in saline, and this is what you see here. So the exosomes that we spun down produced a red light, a strong red light, saying, okay, what we engineered here is really exosomes. So the answer to this question would be, yeah, these MacT cells we can genetically engineer in a very efficient way. Just for your reference, 130,000 G is a crazy force. I mean, pilots go through training. I think they have a G force of 8. So I think it's crazy. I mean, what you need to apply in order to precipitate these exosomes. Okay, so your question now might be, okay, how does this red bulb help us? And we want to get SYNGAP into the brain, not red light. My answer would be, yeah, that is definitely true, but we do use, want to use this IRFP, this red fluorescent protein, as a tracer. So it makes it very easy for us to see where these exosomes go in a mouse. And you have actually seen data based on this technology before. I mean, you've seen this slide. That was actually mice, exosomes, sorry, that had a CD47. Remember the don't eat me signal plus this label. And again, repeating myself, there was this almost 60% decrease in exosome uptake by macrophages. So very efficient as far as don't eat me is concerned, but also very efficient as far as this label is concerned. Okay, now the strategy to be used in this SYNGAP research project. We will create exosomes in MacT cells that are labeled with both RVG, that is this is the brain shipping label, the CD47, the don't eat me signal, and our label, the light bulb, so that we can track these exosomes in a mouse. So the mouse will actually have this part of DNA in a circular form, actually, but this DNA inside. Again, IRFP for assessing distribution and expression of our genes, the RVG, brain homing, CD47, don't eat me, and then we will use a technology called PCR to assess the expression of SYNGAP in the mouse's brain. The original plan for this proposal was to try oral administration in our mice. Then in hindsight, I started wondering why we wouldn't even supplement this approach or change the approach to nasal administration. There's something like precision olfactory delivery, these little spray units, and you actually deliver any medication, including milk exosomes, into the nasal cavity, ideally in the upper nasal region, because then you're just one step away from entering the brain. This is olfactory bulb. You just have to cross, milk exosomes just have to cross a tiny layer of cells and right in the brain, compared to oral. You go to the GI tract, has to circle back to the brain. So we will probably try both, I mean, nasal and oral delivery in our research project. So that was the point where I thought I prepared my presentation, and Hans Schlecht reached out to me because I shared this proposal, or the slideshow with the Syngap Research Fund last week, just for you guys to look at. Hans came back saying, well, can you talk about different payloads also? So I figured, okay, I go back to my presentation, change again to include what's on the horizon. So what does the future hold for this research? That allows me to address a couple of more questions before actually talking about what Hans was asking. So the questions that we are facing with cell cultures like MacT is, isn't exosome purification from cell cultures very unproductive? So the question being, do you get enough exosomes? And the answer is, yeah, that is a challenge, because these cells just don't secrete a whole lot of exosomes, enough to do experiments in mice, but definitely not enough to this large-scale human research study. And I'll come back to it in a minute. And then the other question may be, why do you want to lock in on RVG and CD47 in your cell lines? And I think that's a very good question, because RVG will take these exosomes to the brain. A person suffering, let's say, from pancreatic cancer might want to have exosomes and drugs delivered to the pancreas. So we don't really want to lock in on certain modifications. And CD47 works great, but there's alternatives. So we're also playing with another protein called CD24, which has the same efficacy as CD47, and we will probably try both in combination to see if there's some synergistic effect that decreases macrophage uptake even more. So now we do have a really big grant pending review by the National Institutes for Health that is targeting or designed to develop what I refer here as a universal code. So this code would allow us to change the shipping labels and any surface modification, like shipping label or CD47, really in a heartbeat. So we collect the exosomes from these codes and just put our markers or proteins on there. And so this is a very universal, very flexible system. It's always very scalable, because these codes produce lots of milk, as opposed to milk exosomes, as opposed to cell cultures. And the reason why we named codes in that pending grant application is you can create a transgenic code in 2 or 3 years. The transgenic cow takes about 8 years, and the grant application covered a period of 5 years. Obviously, grant reviewers would not like the idea of us proposing something that we know a priori is not going to work within the funding period. So this is what we call our universal code, because it allows to do any zip code on the exosomes, any payload. And then the bad news for you is my intellectual property people at the university told me, well, you cannot disclose the specifics because it will compromise the patentability of this invention. So, this code hopefully will be out there in the future, but I cannot exactly tell you, or cannot tell you exactly how we actually change the zip codes on these code milk exosomes. The question that Hans Schlecht was asking is really, is there any payloads? Payload is what you put into these exosomes, other than mRNA or DNA that you could actually use in these modified exosomes? And the answer is yes. There's at least 2 other strategies that you could use. So this is the anti-exosome So you could, in theory, I'm going to show you two, use small interfering RNA, which is really tiny snippets of RNA that turn off genes. So in other words, tells, in your case, the SYNGAP would tell this dysfunctional copy of the gene to shut up, basically. Yeah, don't make what you're doing, because even these non-functional copies, they do something. So they produce certain metabolites, and there's diseases where these abnormal products are really contributing to the disease. I don't think anybody ever looked into this possibility in the context of SYNGAP, but for many other diseases, this is a problem. This is actually the main problem. So you want to eliminate the dysfunctional copy. The second alternative that I was going to share with you, that's getting really, really complicated in the lab, not for this talk, would be the use of CRISPR-Cas, and that's a gene editing tool discovered not too long ago, which allows you to repair dysfunctional genes. So in other words, you cut out a certain section of the gene and repair it with the normal gene, and then all of a sudden you have two normal copies in the brain. Again, this is very involved because all of a sudden you have to load these exosomes with proteins, with different types of RNA and DNA, but it's something that is in progress, and that's why I wanted to share it with you. Okay, so now I want to acknowledge my funding agencies, people in the lab as of last November. So your group is named right here, the Syngap Research Fund. Really thank you a lot for your support. And this is collaborators and lab members since my lab got into this exosome field. So that was dating back to 2013, 2014. Then finally, an acknowledgement to Hans Schlecht. He actually was the one who introduced me to SYNGAP, reached out to me, and that's how this whole story took off. So I want to make sure I acknowledge him for his valuable introduction to the Syngap Research Fund people. With that, I think I'm at a stage where I'm ready to take your questions. Thank you.
Wow, thank you very much, János. That was a tour de force, and I really appreciate you making it so accessible. I realize it would have been easier for you to just plow through some existing scientific slides. So thank you for spending your time on that. Sure. Since you had that wonderful picture of Hans up, I'm going to invite Hans up to ask some questions. But before I do that, let me point you to the Q&A. Do you mind, János, stopping sharing your screen so people can
sure.
see us more than the? Thank you again. All right, this is great. So while I'm trying to figure out how to bring up Hans, if you can find the Q&A, there's a number of questions. The first would be from one of our volunteers saying, would there be any side effects to this mRNA treatment, given that the COVID vaccines also had some? I mean, you talk about, I mean, I realize that's a bit down the path, side effects in humans, because right now, just to anchor the audience, what we're talking about is proving concept that you can deliver to the brain and affect SYNGAP expression in a mouse. And if we got that, the next step would be to say, okay, where do we go from here?
Yeah.
But do you want to talk to the?
Yeah, side effects. Yeah, I mean, I think I touched upon it a little bit in my presentation. It's a good question, so I really appreciate that. But side effects possible, yeah. I mean, because you would maybe express SYNGAP in a tissue that it's normally not expressed in, and you don't know what that does. We try to minimize side effects by the strategies that I shared with you with the homing peptides. So we really want to make sure the vast majority of the SYNGAP DNA, so it's, in our case, it's a plasmid, it's not mRNA, it will produce mRNA, but the vast majority of our RNA or DNA will go to the brain. But yeah, so remains to be seen, I guess, is the answer. So we will have to just monitor a situation and see if there's any adverse side effects.
Great.
It's a fair question, a fair answer, I think.
Yeah,
we don't know yet. Yeah.
And Hans has joined us. Hans, do you mind if I just let Dr. Zemplani play through the rest of the Q&A? And before I forget, I do want to echo what János said about Hans. SRF is lucky to have someone who is so obsessed with science and our children and going out there and hunting and finding people like Professor Zemplenny. Because as I think as the audience is gathered from the presentation and that impressive list of funders, a $100,000 grant from SRF is not changing the fate of the Zemplenny lab. We are fortunate to have this work being done. All right, Marcos from Germany. If the milk exosomes is used for transportation of mRNA, how much will cross the blood-brain barrier? Are there numbers from former studies? Again,
yeah, it's a very good question, and we have a very rough estimate about the oral bioavailability. So it's not a brain. So oral at the minimum, as of today, we say 25% of the exosomes are absorbed. Crossing the blood-brain barrier, we do not have any quantitative data. We do know it ends up in the brain, but we don't know the percentage or the mass that enters the brain.
And then there was a question about, was the milk raw or processed? I think you spoke to that a little bit with one of your slides.
Yeah, so it really doesn't matter. I mean, in our case, most of our milk studies, the cow's milk study, we do with milk from the grocery store, so it's processed. We did some work with raw milk, either from cow or from human milk donors in the nutrition arena. But yeah, typically in our lab, we use processed milk from the grocery store.
Great. And then Ilya hit you with 3 questions, and Ilya is a friend of the fund with a child with a different disease. So you can see where he's going. Is there any way to limit where exosomes are expressed, much like AAV when we use promoters? How easy is it to pivot to other genes? They're already signing up for more work. And lastly, could we package more complex constructs where we knock down a mutant via shRNA or similar and overexpress a gene?
Yeah, well, maybe let me start with the third question. That's the easy one, because I touched upon it at the very end of my talk. Yeah. Actually, I have done siRNA a lot and small hairpin RNA, and that works like a charm. We do this more in the context of brain cancer research now in my lab, where we turn off certain genes, kind of what I showed with this smiley face, turning off, telling this one copy that is dysfunctional to stay silent. So that is definitely an option. CRISPR-Cas, I think, yeah, there is lots of talk about that technology in milk exosomes or in exosomes in general. But again, then packaging becomes an issue, because you will have to get everything, I mean, all these components and building blocks in one single exosomes. And that's also a problem when we do our analyses. I mean, you measure, when I talk about the palette of exosomes, we're talking about trillions of exosomes. And then there's a certain percentage that will have one cargo, another percentage that will have a different cargo. As a data piece, it looks fine, because it says, oh yeah, we find everything in there, but it's not like everything is in each and every exosome, which is what you would need. And that becomes a little trickier with the CRISPR-Cas. We could engineer some of the components into our milk exosomes, but then, I mean, the specific guide strains and all, that would be a little complicated, but it's something that we're working on. So it's a very good question. Yeah, other genes, absolutely. I mean, yeah, anything that you want to restore as far as expression is concerned, or knock down as far as siRNA is concerned, we can do that.
And then is there a way to limit where exosomes are expressed, much like AAV when we use promoters? I think the mailing labels we talked about, right?
Yeah, that's also a little challenging. We do have a transgenic mouse in which we knock down the expression of exosomes, but to date, there's not one single gene that you can knock out and inhibit exosome biogenesis to 100%. So the gene that we work with knocks down exosome biogenesis by, I think, 80%. So you still have some residual exosome production in these tissues. So that's, I think, the challenge. I mean, technically it's possible, but I think that gene has yet to be discovered. And that is the gene that we would actually have to rely on if we want to really repress exosome or restrict it to a certain tissue.
Hans, this next one's yours.
Yeah, so Janos, fantastic to have you involved with SRF. And this is a project where I have to manage my expectations because I'm that sort of hopeful. And one of the questions, we see host proteins make it into, HIV particles that bud off an infected cell, exosomes that come off the intestinal mucosa can take a hepatitis E virus with them or an enterovirus with them. Are there other host proteins that are making it into these exosomes? And could they be immunogenic?
Mm-hmm. It's a good question. So I don't know I know that you had to some other events to this morning. So at the very beginning of my talk, I talked about some of the cargos in exosomes. And there's lots of proteins in exosomes. And you're absolutely right. In theory, that could elicit an immune response. What we have done in human studies, and I think that's probably very helpful in this context, is to study the immune response in humans after exposure to milk exosomes. And we did this both in vivo, I'm basically just giving exosomes to human subjects, but also after isolating immune cells from human plasma, like PBMCs, and culturing them in the absence or presence of milk exosomes and measuring cytokine storms. And milk exosomes are really very non-immunogenic, at least in this model and these model systems that we used. But we have to see once we really, enhance the accumulation in the brain, if there's any issues with that.
And it's a little bit tangential or to the side here, but pasteurization, as I've seen in your publication record, it disrupts the normal exosome component to cow's milk?
No, you remember the paper, correct? I talked about this very, very early on when we talked about stability of exosomes in today's presentation. And it's typically the homogenization, the shearing of exosomes. And the reason for that is you apply the shear force and you actually disrupt the membrane. The pasteurization, the heating had actually no effect. You can pasteurize the exosomes and you can do fat adjustment and any other processing and you don't lose the exosomes. It was really just the homogenization that caused, I think, a 50% loss of exosomes.
Okay. It's fascinating work. Absolutely. Thank you.
Okay,
so the next question comes from Dr. Elizabeth Heller, who I'm thrilled is here with us. She is a member of our scientific advisory board and a grantee and an aunt to a Syngapian. So we are very grateful for her and her many roles in this community. But she asks, can you explain the rationale to make Syngap exosomes in vitro versus in cow goat? Is there any benefit to trying both strategies at the start?
Yeah, well, potential benefit for sure. But as I was trying to say in my presentations, if we wanted to make transgenic goats or cows, it's crazy expensive. This project proposal I named, or I mentioned this with the universal goat. I think it's a $5 million proposal NIH, and with high risk. So it's that project can fail. So this submitted to a particular program at NIH. Sorry for getting expanding my given extensive answer there. So it's NIH Director's Transformative Research Award. It's big money, and it is for high risk, high return research. They're saying, well, we know 99% of this is not going to work, but if it works, it's a game changer. That's what this universal goat is going to be about. And yeah, goat versus cow. I explained my rationale. The goat you can do, transgenic goat you can do in 2-3 years, a cow 8 years. Do this in parallel for this particular project is just not feasible. And that's why the MacT cells are such a nice model, because we can quickly engineer these MacT cells. The exosomes are basically milk exosomes, and we can make any modification that we want in a rapid way, rapid fashion. Cheers. Gotcha. Thank you. See that. Thanks.
Okay, and And Dr. Heller, if you want to come up and ask more hard questions, please just message me. Suzanne Jones, thank you for this amazing work. Back to the small interfering RNA. Are you considering looking into that methodology more fully in the future? Seems like a viable solution for missense patients. So I don't know, Janos, how close you are to our genetics, and Dr. Heller will be better to speak to this, but broadly speaking, 2/3 of our population have protein truncating variants, so they're classic haplo. And I'd say another 20% based on our registry are missense mutations, which are, it's less clear what's going on. So I think that's what's behind this question.
Yeah, in this context, I think it would be really good to know if these mutations cause, the production of metabolites that contribute to the phenotype in SYNGAP patients. And as I was saying in my talk, I'm not a SYNGAP expert, so I don't know if these missense mutations cause anything or cause the production of some metabolite that contributes to the disease. If that is the case, then yeah, knocking down or knocking out that particular allele would be perfect. It would be much easier probably than what we are trying to do here.
Okay, and then Ilya is back with another question. Is there a packaging size limitation?
Yes, there is. So, I don't know how large a molecule can be, but again, these exosomes have about 100 nanometers in size. There's a physical limitation what you can get into these exosomes. Our loading technology is very effective. Actually, we use very old-fashioned strategies, actually just ethanol and calcium chloride, very basic molecular biology from the old days, and it works great. I think we get about 40%, 50% loading efficiency with this technology. But there's alternatives. There's electroporation and then transfection. So, but we are happy with what protocol we have.
And then last question in the chat is from Marcos. What is the timeline for this study, supposing that what we're after here is proof of concept? So yeah, so the timeline, this is a 1-year project. So yeah, and that's my timeline. Wonderful. Of course, I'd be happy. I'm sure, hopefully, if this turns out well, if we really show proof of concept, I'd love to continue working with your group after this 1 year. But I think this is a good first step. Just saying, well, is this leading to something? And then we can discuss again, I guess. Yeah, we would love to see that. Hans, you got more?
Janos, so are there preclinical examples where exosomes have modulated the phenotype of an animal or iPSCs, let's say, and their output? Well, if you go to the FDA registry or NIH registry of clinical trials, there is an increasing number of exosomes, not necessarily milk exosomes, but exosome trials for delivering, it's typically siRNA, and then increasingly now, at least at a level of talk, for the CRISPR-Cas technology. But I don't think this has ever led to an approved drug or treatment. So this is all trials in progress. Yeah, it seems like lipid nanoparticles are very in vogue right now. Yeah, exactly. Yeah. Okay. That's one more question. Right?
So yeah, JJ, who is asking an interesting question. What about human breast milk? Could human milk express enough exosome without the immunogenicity problem? Yeah, that's a very good question. If you have the donors willing to give enough milk and extract enough exosomes, that actually might help if there is an immunogenic potential. Like I said, we have no evidence to suggest that cow's milk exosomes cause a problem in this regard. But if they do, yeah, human milk actually not a bad idea. So one could try that. But again, then you run into the issue. Well, if you want to genetically engineer these exosomes like RBG or CD47, that is, of course, not an option with humans. So I think that's where the cow's milk is, or the goat is really the way to go, because you can really engineer them any way you wish. May I ask you one sort of broad question that isn't specific to your presentation? One of the things, when I started SRF, we talked to a lot of scientists, and I heard a lot of things I didn't understand and I don't remember. But one thing someone said to me that really stuck with me is, if you want to know where neuroscience research is going tomorrow, look at where cancer research was yesterday, because there's so much more money in cancer and so much more science that, you know, you can get a sense of the future in neuroscience from looking back a little bit. And so I know that some of your work is being used, as you mentioned at the top, in brain cancer. Without, I mean, you probably talk about this for days, but can you give us a sense of how to think about where this might, without making any promises and recognizing that we're just talking about a proof of concept? Can you just paint for the audience broadly where exosomes is going broadly based on what you've already seen in the cancer space?
Yes, I mean, sure.
Sorry, it's a big
I think, yeah, it is. But yeah, I mean, first of all, I agree with you. I mean, yeah, cancer, if you look at what happened to cancer yesterday, you get a sense for where the field overall is heading. And then I'm really coming myself from, well, origin from nutrition, but as far as drug delivery is concerned, our first grand applications are all in the brain cancer field. And yeah, then I didn't even think neurobiology until Hans reached out to me saying, well, I mean, this is a cool technology, and then maybe, and so yeah, I mean, the answer is yes. Where this is going, I don't know. We have to see if we can really restore gene expression to normal levels in the brain, or if, like you said, with the missense mutations, maybe it's good enough to eliminate these missense copies. I don't know. I mean, that's something that we definitely would have to look into. But it's an exciting field, and if you keep up with exosomes in general, it's almost like the microbiome. There was just a few papers and publications a few years ago, and now there's hundreds, if not thousands per year, and not all of them are about drug delivery. Some of them are nutrition and just cell-to-cell communication. I mean, it's a crazy field. I mean, it's very, very busy these days.
Yeah, and we're fortunate that hopefully this proof of concept will get SYNGAP in on that rising tide. So once again, thank you from all our families for taking this project on, Dr. Zimplenny, Professor Zimplenny.
Absolutely, yeah. No, it was a pleasure talking with you. I was not lying in my opening statement how much I like your group.
Thank you. It's very kind of you to say. All right. With that, give everyone their day back. I appreciate your time, Professor. Thank you.
Thank you. You have a nice day. Bye.